Hey students, this is your presentation on meiosis, the formation of sex cells. So if we look at what is meiosis, it's just a special type of cell division, much like mitosis, that results in 4 sex cells or gametes with just half the number of normal chromosomes, or half the normal number of chromosomes or haploid. And it is a process that ensures that sexual reproduction happens correctly. Keep in mind that uh, during sexual reproduction, or those organisms that go through sexual reproduction, half the chromosomes come from mom, half from dad, and that when they join together, they form a organism with a full set of chromosomes, a diploid number. So, again, meiosis is to ensure that each parent is donating half the amount of genetic information. So, what is the point? It's again to make gametes that are haploid, and this is to ensure or to maintain that the chromosome number is correct throughout the process. It's also to create genetic diversity, or a result of making gametes creates genetic diversity. There's a couple of things that happen through this process that are a little different than mitosis that result in being genetically different. Things like independent assortment and crossing over to random fertilization is what we call it, really result in really none of us being the same individual, right? And ensuring that although we're similar, we're not, all of our genes are not identical. And that's how we kind of get siblings, where you might have some of the similar characteristics with your sibling, but you're not identical. So again, these are the 3 ideas of how meiosis increases genetic diversity through sexual reproduction, that random fertilization piece I was talking about, the independent assortment, and the crossing over. So if we look at sexual reproduction during fertilization, 2 genetically different cells combine to make a zygote. Here we have the egg and the sperm. When the sperm fertilizes the egg, we get a zygote, and then, of course, we get the embryo in the end. And if you look at the number here, again, each chromosome, or each parent cell, parent gamete, has half the original of the total. So when the egg and the sperm join together, you get a diploid number. Anything other than that, either too many or too few, is going to result in a genetic disorder. So the fact that, again, we go back to this, it's called random fertilization. So during sexual reproduction, there are thousands of sperm that are trying to find the egg. So which one of those sperm, with whatever type of genes it has in them, is completely random in fertilizing that egg. And which egg is released, because every egg is a little different as well. So it's just a random fertilization. It's completely by chance that the way we are, genetically speaking. So the fact that, again, this during the fertilization process, whatever egg and whatever sperm are actually used for fertilization, it's just a completely random set. So that aspect of it creates some genetic diversity, because each one of these 3 sperm here in this picture are genetically different, although they share some of the same types of genes. They're all genetically different, right? So independent assortment. So, again, humans have 46 chromosomes, all right? That is the diploid number. Every single one of your cells has 46 chromosomes in it, and chromosomes occur in pairs, 23 pairs, or 46 total. And that's because, again, you get 23 from mom, 23 from dad, to make 46. So, excuse me, those pairs are called homologous to each other because they're similar in size, shape, and types of genes. It just is really dependent on which type of, within the gene itself, what type of gene there is, whether it's the dominant gene or the recessive gene. Right? That's where we get into those combinations. But it really is kind of by chance when the chromosomes are lined up to be split, when they're copied in meiosis, at the beginning of meiosis, and whatever way that they are sorted into which cell is completely random. And that's kind of what independent assortment says, right? When the chromosomes separate in meiosis, which way they're separated is completely random. So the third part is crossing over, and when in meiosis, what you've hopefully seen in the lab, the simulation, that at the very beginning in prophase one, you have these homologous chromosomes that pair up to each other, and because they get so close, oftentimes the DNA kind of gets entangled. The DNA kind of switches. So that just results, when the DNA switches, in just different types of genes. So here we have the picture of the chromosomes lining up, and here we have a picture of them crossing over, and in the end you have two chromosomes that are just a little bit different, genetically speaking, than the start. So that's crossing over. It occurs in prophase one. And because we get different combinations of genes, that of course leads to different combinations of traits, and that's what makes us similar. So you might have, or different, that you might have some similar genes, of course, being related, but you'll be, and that's, again, siblings may have some similar features, but they're a little bit different in the end. So let's do meiosis. All right, let's draw some pictures. Make sure we get everything down. So here in your notes, whether you do colors or not, I think it definitely helps with multiple colors. But here, the difference we just want to highlight here, male meiosis and female mitosis is just a little different. So in males, we have, it's called spermatogenesis. So in the end, we start with one cell and we go through 2 rounds of division to ensure that those cells are haploid. In females, it's just slightly a little different. It's called oogenesis, and it produces 1 egg and 3 polar bodies. And the reason is the other 3 polar bodies kind of shed all of their cytoplasm into this 1 egg, because you have to understand, again, that during sexual reproduction, the sperm has to find the egg. So if the egg has a greater surface area, it's going to be easier to be found. So it's important that these eggs are just larger in volume than these sperm. Okay, so in female meiosis, called oogenesis, you get, in the end, 1 egg and then 3 polar bodies. So spermatogenesis, again, here's the picture. You start with 1 cell and you end with 4. And in oogenesis, again, you start with 1 cell and you go through, and you can see kind of all the cytoplasm just kind of goes into 1 cell, and in the end you get 1 large egg and 3 polar bodies that are discarded. Non-disjunction, excuse me, non-disjunction. When the chromosomes line up in the middle, and I'm gonna stop here and kind of go back and just so we know what we're doing here. So this picture here, I'd like to go over with you. So if you have your pencils out, you want to give it a shot, that's fine, but I certainly want to review this for you. What I'm doing for the rest of this presentation is kind of going over some of the terminology used in this process. So let me get back to some other terms. Non-disjunction. So when chromosomes don't separate correctly, that kind of results in a different number being represented in the resulting cell. So for instance, in metaphase, when the chromosomes line up in the middle, sometimes they don't separate correctly. Sometimes a few more go to one side, and then the other side is left with too few. And so that results in either having too many chromosomes or too few. And this is what results in things like Down syndrome, which is 47 chromosomes. We're going to look at this aspect during an activity called karyotyping. But I just want to throw this term out there that sometimes the chromosomes don't separate correctly, and that results in either too many or too few chromosomes in the cell. And again, just having 1 extra chromosome or too few, just 1, results in something that's genetic disorder. So again, here we have sort of the picture of how that happens. Here we have in meiosis 1, the chromosomes should separate evenly, but it doesn't. It doesn't. You have too many that go to one side in the resulting cells, and then you're left with too many in the end. Okay, and it's all about chromosome number. And meiosis is specifically designed to ensure that each cell has the right number of chromosomes in it, but sometimes it doesn't occur that way. And that's what non-disjunction is. And that's how something like Down syndrome occurs. If then that cell is used for fertilization, because again, it's all random which cells are actually used for fertilization or participate. If that egg or that sperm happens to be the one that is fertilized or used in fertilization, then you end up with a genetic disorder. So a karyotype, last thing, a karyotype is a picture of someone's chromosomes. And we're going to do a little review on this next class, but it's just keep in mind humans have 46 chromosomes, so oftentimes scientists can take a picture of all 46 and kind of line them up in a map. And it's a way that doctors can really determine if something had happened in meiosis, if there's a genetic disorder, if one of the pairs, because again, chromosomes occur in pairs because you get one from each parent or half from each parent. If there's something wrong with one of those pairs, scientists can really determine whether or not there's a genetic disorder. And oftentimes they can do this in the fetus, right? Oftentimes parents know that their child is going to have Down syndrome prior to the baby even being born because you can do a karyotype of that child while it's still in the mother. So, again, a karyotype is just a picture of the chromosomes. And we're going to talk about these two terms here, sex chromosomes and autosomes. An autosome is all the chromosomes that do not determine your gender. Sex chromosomes are. So sex chromosomes, you're either XX or XY. XX is a female, XY is a male, and they determine your gender. So if it is a chromosome that doesn't determine whether you're a male or female, then those are called autosomes. And we'll kind of work with that in the lab. Right? So again, the purpose of the karyotype is to show a picture of all the chromosomes, and in the end, you can determine that not only the gender, but chromosomal disorders. So, well, we're not going to do that, but we are going to stop and draw some pictures of meiosis.